1 or 2 just to get a clean cut, and then you can get started. Alrighty, starting in 5, 4, 3, 2.
Hi, everyone. My name is James So. I am an assistant professor at Stanford University. And I'll tell you a little bit about some of the recent work that we've been doing in developing computer vision algorithms to study genomics, and in particular to study genomics across different spatial variations and also across temporal variations. And this also will be an opportunity to give a tutorial of how we can actually leverage some of the recent advances in deep learning for computer vision. Great. So to set the stage for this presentation, right? So I want all of us to do a thought experiment together. So imagine if I've given you a list of descriptions of an individual, right? So here I've given you a list of descriptions, which could be taken from a medical record or from clinical notes. And what I like you to do is to try to visualize in your head as much as possible of what the face of this person looked like. Right, so take a couple seconds to do that. Okay, and now I'm actually show you the actual face, right? So this is the actual face of this individual. And you can see how well the vision in your head, whether it agreed well with this image or not. Right. So the faces of this individual is actually very interesting. Right? This is taken from a New England Journal of Medicine article from a few years ago. So it's actually the face of a truck driver. And one thing that's quite notable is that this truck driver, the face is actually quite asymmetric. Right? So you see one side of the face is actually much more wrinkly and sagged compared to the other side. And this is really because of the exposure to the sun, right? When the truck driver is driving on one side and the window is closer to that side of the face. And it's a really nice image because first it captures quite vividly, right, this interaction between the environment and between the environmental exposures, between the underlying biology of this individual and how this all affects the health status and disease outcomes of this individual. But it also captures, I think, a really important point, which is that there's actually a lot of information, very rich information in the images, right? And that's actually very hard to capture with with the standard text descriptions, right? Because if we actually go back and look at the text, right, just based on this, my vision of what this person looked like is actually very different, much less rich and vivid compared to this actual image. Whereas the text would maybe have a few bytes of information, right? But whereas the image itself could actually contain megabytes or even gigabytes of information. And this is the case for images of humans, of faces, something that we're quite familiar with, right? And the problem is even more stark and even a bigger challenge if we're thinking about how to describe objects that are even smaller. Right, so here I actually have an image of a tumor biopsy from a breast cancer tumor, right? And this is actually just, and on the right here, we actually have a individual cell. It's a microglia, which is kind of an immune cell, which I'll come back to in a little bit. And here we have the motion from a microscope of this individual cell. Right, so how would we even describe these objects? Right, so that's actually quite challenging. And the reason why it's challenging is because it's in English, we really lack a very precise vocabulary with which to describe the phenotypes and morphologies and dynamics of these objects, right? It's hard enough for us to even precisely describe faces of individuals. It's even harder for us to describe these smaller objects like biopsies and individual cells. And this is really the motivation for this talk and for a lot of my research, right? When we think that the recent advances in computer vision, right, the goal is to really use those computer vision technologies to enable us to more deeply and more precisely phenotype these objects that would be hard for us to describe otherwise. And in particular, we want to use computer vision and to use these deep learning algorithms to basically create a new vocabulary, a new language of morphology. And this language actually then describes objects across different physical scales, from the scale of individual cells to that of humans and organisms. A lot of the work in my group is actually in developing these computer vision algorithms, right? So, and and many of these are actually also widely used by some of the companies that you're familiar with. Right? And a lot of the really advances in computer vision recently is based on advances in sort of artificial neural networks. Right, so I'll give some more examples of this throughout the talk. But let's say if you wanted to build, like in this case, like a self-driving car, right? Then you would like to have the car to be able to look at the surroundings and to identify what are other cars and what are other individuals, right? So you should be able to detect these different objects and to characterize what they are. And in a nutshell, right, the kinds of algorithms that people currently use to detect and to classify these different objects using computer vision and using self-driving cars are really based on these different layers of convolutional neural networks. So it's a particular type of a deep learning algorithm. And what these algorithms are doing, again, this is just an overview, which I'll come back to in a little bit, is they're basically scanning across this image, right? So let's say if you're trying to detect the particular car, then the convolutional neural network will scan across this image, right? And so each column here corresponds to one layer of the convolutional network. So the initial layers, right, so these are the columns to the left here, are looking for local objects, right, in this car. So maybe it's looking for wheels, or it's looking for windows. And some of the later layers, so the columns that are in the middle or to the right, are looking for higher level or more abstract concepts, right? And these layers come together in the end to come up with a classification, which algorithm then classifies with some confidence whether this is a car or a truck or an airplane. So the goal of this talk is to give you a tutorial and to illustrate how we can apply these algorithms instead of looking for cars and for computer vision tasks in the world. How can we actually use these technologies to really help us to understand spatial variations in genomics, as well as temporal variations, temporal dynamics in individual cells in a way that's directly relevant for human diseases. Okay. So how do we actually think about using these vision technologies for pathology, right? To understand spatial heterogeneity and genomic heterogeneity. So the setting that we're working with here is actually in histology, right? So here's just this. Here I'm showing you on the top left is a standard kind of image, a histology image taken from a breast cancer patient. Right, so these are kind of the routine image that people commonly generate, and the pathologists would often look at these images by eye, right, and then make some assessments and diagnosis of the stages or the of this patient. All right, so on the bottom left here, we actually have the clinical annotations by the pathologist of where in this image he or she believes to correspond to tumor regions, and where it actually corresponds to normal cells, normal regions. Right, so these histology images are widely used, and they're very powerful because it allows you to really capture the spatial heterogeneity, right, of different cells and different parts of the tissue, different parts of the tumor in real time and in real space. In parallel, as many of you know, so there's also a very powerful set of technologies based on genomics, and more recently based on single-cell genomics, which captures quite a complementary orthogonal types of information. Right, so typically what people do here then would be they would take these biopsy samples, they have to dissolve the cells, right, to extract the individual single cells. And once they dissolve and extract the individual cells, then they can do single cell, for example, transcriptome measurements or single cell sequencings to capture the expression profiles of those individual isolated cells. So these two different technologies both have pros and cons, right? So the advantage of the single cell or of the genomic analysis is that first, you actually have much richer information, right? For every individual cell, if you do RNA-seq, you actually have a lot of the genes, thousands of genes that are being measured. The disadvantage, the downside, is that you actually lose the original spatial information, right? You don't know which location in the tissue this cell comes from, so you lose actually the bigger context. The advantage of the histology imaging, this current pathology workflow, is that you do have the spatial context, right? Because you have the original tissue, but the measurements you see are fairly limited information, right? Maybe you see this is normal or this is normal or malignant, but you don't actually really have the information about all this rich gene expression of thousands of genes for these images, right? So there's certainly a trade-off between the two. And what we have been doing recently is to actually use computer vision to develop new technologies that allow us to get the best of both worlds, right? So you have both the spatial imaging data, and at the same time, you also have rich genomic readouts that are spatially resolved. So here's just an example application of our algorithm, which we call the STNet algorithm. So here's the input example, right? So we have the histology image that we saw before from breast cancer patients from a biopsy. And the STNet, our algorithm, is able to impute the computationally synthesize spatial variations in gene expression profiles, right? So for example, let's say if you're interested in this gene FASN, which is one of the breast cancer biomarkers, then the STNet would be able to impute the expression profile for FASN on top of this histology image. It will tell you the parts in yellow are the places where the FASN is highly expressed, and parts in blue is where this gene is lowly expressed. And if you're interested in a different gene, right, here I have a second gene here, then the algorithm would also synthesize and instantaneously impute the gene expression profiles for the second gene. And with this algorithm, right, now we can actually impute the expression profiles that are spatially resolved for over 100 genes. And we have experimental measurements, right, actually using these spatial transcriptomic technologies, which I'll describe in the next slide, to validate the computations from the algorithm, and they're actually in very good agreements here. Right, so this is done on one of the test patients. So just to make sure everything's clear, right? So the second column here, these are computational predictions that are synthesized gene expression profiles directly from the histology image, right? And then on the right here, right, the third column corresponds to the experimental measurements that we can use to validate these computational predictions, and they're in good agreements. And if we have a different patient, right, if you have a second patient, right, maybe you have a different biopsy sample, a different histology image, that's also fine, right? So the algorithm would take a look at just this raw histology image and is able to actually make profiles, right, for this gene expression of FASN and also for 100 other genes. And that also agrees quite well with the experimental measurements, right? And this is also quite interesting here, right, because this image itself, but was the standard clinical annotations, the pathology would actually say that, oh, this entire image is almost uniformly made of tumor regions. Whereas the algorithms is able to detect that even within all of these tumor regions, there is still substantial spatial variation in the expressions of these key tumor genes and these key markers. And that can be very useful for more fine-grained and more precise prognosis, as well as for treatment planning. So this work was recently published, right? And you can find the paper detail here. It's published a couple of months ago in Nature Biomedical Engineering. Okay, so now I want to take a few minutes to explain a little bit more detail of how we actually develop this algorithm and how do we validate it and apply it, right? So to train the algorithm, right, we actually need to collect actual experimental data that are spatially resolved transcriptomes. Right? And this is actually done in collaboration with my fantastic collaborator, Joachim Lundberg, who is one of the pioneers who developed these spatial transcriptomic technologies. So how it works in a nutshell is the following. Let's say if you have a breast cancer biopsy, right? There's a tiny little biopsy. And what you do is that Joachim has developed this chip, right? So I have an example of this chip here. You basically overlay this biopsy tissue directly on top of the chip. Now, what's special about the chip that Joachim developed is that you actually have little probes on the chip, right? So these are little dots here in our cartoon. And each probe actually corresponds to a set of basically barcoded needles, right? So think of each probe as corresponds to like a zip code that tell you where's the XY location of that probe. And what happens when you overlay the tissue on top of this chip is that these little needles will basically puncture and get into the cell, right? And once it gets into the cell, if it actually detects some transcripts in the cell, right, it would actually then attach a barcode, the spatial zip code on top of the transcripts. And the beauty of this is that now when we actually sequence the cells afterwards, now we know exactly where the transcript come from, right? Because we have the barcode, the zip code attached to the transcript. So that's how we can experimentally measure that for this gene FASM, right? That's where here are the regions in the middle where it's highly expressed, and the surrounding areas where it has low expressions, whereas for a different gene, XBP1, you have different expression patterns. And this is quite high throughput in the sense that we can actually do this and then be able to measure expression values of several hundred to thousands of genes in parallel. Okay, so this is the training data that we generated to train and to validate our algorithm STNet. Right, and STNet itself, it's actually very similar to this kinds of image recognition or self-driving car kinds of computer vision algorithms I showed you a few slides ago. Right, so in a nutshell, what STNet is doing is that it's also scanning across the individual image. Right, so it's looking at local patches. I've shown you some examples here. Right, so each local patch is around 100 to 150 microns. Right, so it's quite small. And the STNet algorithm scanning across those patches, it learns what are the important features automatically, what are the important features in each of those patches. And that's what we're calling this latent representation. And once it learns that representation, then it uses that information to impute the expression profiles of hundreds of genes, right, at each of those locations for each of the probes. Right, so this is the algorithms trained. And with this, it's actually able to impute the expression profiles of over 100 genes, right? And it's able to do that at resolutions around 100 microns each. So it's actually fairly high spatial resolutions. So this was done on breast cancer patients. And we actually did a lot of rigorous testing to evaluate that the algorithm really works well. Right? So the best way to do testing is to actually really take very independent patients, independent data. So here we actually use data just generated by a different group. It's actually generated by 10x Genomics, so a different company. And they have different ways to collect these samples and even different imaging protocols, different sequencing protocols. So this is actually quite a strong test to see whether the algorithm that we've developed on our set of breast cancer patients, how well it will work on a very different set of data, also from breast cancer patients generated in this different environment, different contexts by 10x. And it actually works quite well, remarkably well, right? So each dot here corresponds to one gene, right? So the X and Y axis corresponds to the performance of the algorithm in predicting the spatial expression profile of that gene on two different test data sets. And so the AUC is basically one of the common measures of how well the algorithm is doing. And we can see here in the top right corner, here I highlighted that in the red circle, but there's actually over 100 genes where the algorithm, STNet, actually is making very reliable predictions as compared to the experimental ground truth measured on both of these test sets. Right, so these are the 100 genes where you have AUC above 0.8, above 0.9. And these are the 100 genes where we think that the algorithm can really quite reliably computationally generate these spatial profiles. And these 100 genes also captures many of the key genes that we're interested in, right? So these include cancer biomarkers, immune markers, as well as genes that are really important for the mobility and architectures of the cells. So the beauty and the power of this kind of approach is that once we train the algorithm, now we can actually do this and apply this to new data or where we just don't have the corresponding experimental measurements. Right, because the experimental measurements, while it's great, but it's actually quite expensive and can be quite time-consuming to generate. Right, so there's still only a few groups around that can reliably generate these high-throughput spatial transcriptome measurements. But the algorithm, once we have developed it, that can be applied potentially to any image, including sort of archival images that labs and clinics have already collected. Right, and we have actually tried this. We applied our algorithm and that's trained on our breast cancer data, and we actually applied it to images from archives, from TCGA, and we also show that they work quite well there. And we can also use the output of this algorithm, right, to quantify a variety of things, including tumor heterogeneity, as well as to the extent to which the immune cells are infiltrating into these different tumor regions, which can be captured from these from the synthesized expression profiles. It's also very interesting to us to see what is it that these deep learning computer vision algorithms are looking at when it's trying to predict the expression profiles of different genes. right? So for example, here we were showing sort of a zoomed-in version of this of the biopsy, and highlighted in the bottom row are the regions where the algorithm STNet is paying attention to when it decides whether each region actually has high FASN expression, right? So FASN, again, is a breast cancer, one of the key breast cancer biomarkers. And it's actually quite interesting because here the algorithm actually learns to focus its attention to relatively a few locations in the input, which of course actually corresponds to these enlarged nuclei in the cells, right? So the parts where it's paying large attention to, then red regions actually corresponds to these nuclei regions. So the algorithm actually learned by itself that if you actually have enlarged nuclei, that's actually symptomatic of high expression for this tumor factor FASN. So this demonstrates actually it's quite powerful to use this technology as also as a way to link gene expression profiles, right, to cellular and tissue architectures and morphologies, which could lead to sort of new and interesting biologies. So the final, I think the ultimate application of this technology that we're very excited about, right, is to really think of this as like an enabling platform, an enabling technology, right? So it's sort of like Instagrams, where instead of putting in sort of filters for cats and for dogs, right? So now if we actually look at one of the original histology images from a tumor, we can actually in real time have the algorithm generate the spatial transcriptome profiles for many of the key genes, and we can overlay those spatial profiles on top of the original image like these filters. This, I think, can greatly enable prognosis and treatment design, especially where the tumor spatial heterogeneity is quite important. And as we've seen, it also opens up new hypotheses of how specific genes, their expressions are linked to changes in cellular morphologies. Great. And in the second part of this presentation, the second story, I also want to tell you about how similar kinds of computer vision algorithms can, in addition to capturing spatial variations, can also capture temporal variations, right? Can capture morphodynamics. This is a collaboration with my fantastic colleagues, Shalin, who's at the BioHub, and Tom, who's at UCSF, and it's work that's led by Michael, who's a great PhD student in my group. So here's the setup: is that Tom and Shalin's group are interested in studying these microglia, right, which are kind of immune cells in the brain. So here's the image that Galina, one of our key collaborators, took, right, of these microglia over 24 hours using label-free microscopy. And what Michael did is that he actually developed a system, right, to automatically track and study the morphodynamics of each of these individual microglia cells. Right, so it first tracks the individual cells, it classifies the different cell types that are involved. And then after that, right, you can think of the algorithm, the computer vision algorithm, is sort of being like a private eye that's overlooking each of these microglia cells, except that it's doing this for thousands of cells in parallel. Right, so for example, if I'm interested in this particular cell here at the bottom, right, so it's in this red square, the algorithm would allow us to zoom in into that cell. Here on the right here is the zoomed in version, and you can see that cell is actually having quite interesting interactions, perhaps even interacting with some neurons. And the bottom here, I can zoom into a second cell, right? If I want to be interested in its neighbor, I can see that this neighbor here is actually, when I zoomed in, it's actually having quite different behaviors, right? It seems to be very active, and it's even spinning around, and even make out some of the subcellular organelles within this microglia. So it's very interesting. They have very different behaviors. And these examples also highlight one of the challenges, right? Because these cells are behaving in quite complex and quite rich dynamics. And as we mentioned before, we don't really have a very precise vocabulary to even describe these dynamics to make it quantitative. And this is where a lot of the machine learning and computer vision algorithms also becomes very useful, right? As a way to learn these representations and to learn these dynamics and learn the vocabulary. So Michael had developed quite a special kinds of deep learning algorithm. It's called a particular variant of an autoencoder. So think of this as a way to take this input, these videos that we saw before of individual cells, right, and learns a way to represent these videos in a way that's easy to visualize, to understand. And by the process of doing that, it actually learns what we're calling like a morphology space, right? So this is like a new representation, a new vocabulary that the algorithm has inferred from these videos. So the power of this morphology space is that if I look at any of the particular cells, right, including this as an example, this cell here that we zoomed in here, right? So the cell actually undergoes quite different changes and interesting behaviors. That cell now actually corresponds to a particular trajectory in this morphology space, right? So as it evolves over time, it actually traces out a trajectory in our representation. And if I look at a different cell, right, that actually traces out a different representation, a different trajectory. And I can look at this for thousands of cells in parallel, right? And these 4 cells here, as examples, actually trace out 4 quite different trajectories. Some of them undergo quite large changes, others sort of stays relatively consistent over time. And this is really powerful because now we can actually look at any cell of interest, right, under our microscope, and we can really have a quantitative way to look at and to analyze what is the trajectory of these individual cells. For this particular microglia data set, it actually turned out to be quite interesting in that there are 2 very clear, seems like different clusters of states in this morphology space. Right? So I'm just calling this blue or red clusters corresponds to state 1 or state 2. Right? And you can actually quite clearly visualize these 2 states by looking at this morphology space. Right? So each dot here corresponds to 1 cell. And I'm showing you a couple of examples of what's representative cells, right, in these 2 states. Right? So in the blue state, so you have these cells that look a little bit like couch potatoes, right? Sort of lazy, they're sort of large, and they don't move around too much. Right? In the bottom here, you have these red cells, an example of a red cell in state 2. It's much more energetic, right? It's moving, it's spinning around, sometimes it's interacting with its neighbors. So it's much more active. And we can also show some of the example trajectories, right, of these 2 cells in these 2 states at this panel here on the top right. Right? So in the blue state, right, these cells, even when they do move, they tend to be much slower, right? The speed is much lower. And if they do move, they tend to move mostly along this long cell axis, right? So it's very different types of motion. Whereas it's clear from our analysis, right, that these red cells in state 2, they're much faster when they move. And when they move, they're also moving in a much more sort of a random walk in all the different directions. Right? So these are the kinds of things that is now very easy for us to quantify and to describe because we have this very precise data-driven morphology space of these trajectories of the cells. And what's really interesting here in this example is that when we actually did single-cell RNA sequencing of these microglia, in the sequencing expression space, there also turned out to be at least 2 broad, very distinct clusters. Right? So I also color them blue and red, so as to be quite suggestive of how they map onto the morphology space. Right? And indeed, we can actually experimentally demonstrate and validate that there is a connection, right, between these 2 clusters in the expression space with these 2 morphodynamic clusters in the morphology space. Right, so we did several experiments, perturbations to validate this. One example is if we actually stimulate these microglia with interferon beta, right? So then the morphology space, they mostly move into this blue state, right? And similarly, in the transcriptome space under the same stimulation, they also mostly ends up clustering around a particular part of this blue state, right? So by doing a set of these different perturbations, we're able to establish a linkage between the morphology clusters, morphodynamic states, with the transcriptome states. And I think that's really one of the really exciting directions going forward is that with these computer vision technologies, as we've seen in both examples, right, we can learn powerful features to represent the spatial and temporal dynamics of the cells. And then we can actually connect that to rich information from the expressions, either from spatial transcriptomics or from single-cell RNA sequencing. So hopefully this talk giving you some examples, right? A couple of examples of how we can really take the recent advances of computer vision to learn about spatial heterogeneity of cells, to also learn about temporal morphological dynamics of cells, right? And really to integrate these ideas with powerful data and readouts from high-dimensional, high-throughput genomics. And I think this is especially relevant, especially promising and exciting as a way to really study human diseases, right? with these multiple modalities of data going forward. We, in my group, we've developed a variety of ways to actually make it easy for people, for biologists and for people who are not computer scientists, right, to interact with these computer vision algorithms. So, if, for example, if you go to gradiohub.com, right, so there's actually a place where we have developed and we put different interactive visualizations and interactive versions of many of these algorithms where people can just interact with the models on their browser without having to download anything. And finally, I just want to thank the students and collaborators again. And a lot of the papers and codes for these different approaches are available on my website. And I also listed the references for each of the works that I described. Thanks for your attention, and please feel free to send me your questions.
Okay, great job. Well, that's a wrap pretty much from here. I will just do some quick editing on the front and back ends and get your presentation ready for the event next week.
Okay, sounds great.
Awesome. Any questions for me before we wrap up here?
So do you need anything else from my side?
Let me check real quick just to confirm with you before you head off. No, it looks like we have all of your information in regard to presentation abstract and learning objectives. So I think we're all set. If anything comes up, I'll definitely reach out. And if you have any questions at all, feel free to reach out to me.
Okay, great. Thank you.
Thank you.
So you mentioned the questions from the audience. They will just be emailed to me from whenever they come through. Is that right?
That's correct. So they'll be emailed to you in real time as they're submitted by the audience. You'll get a majority of questions on the day of the live event, and then there is the 6-month on-demand period where you can expect to receive some questions throughout that time period as well. Okay.
And should I expect them to come from like a particular email account, or will they just come from the individual, whoever asked the questions?
Yeah, it will be a specific email. I can't remember it off the top of my head, but it'll just be an automated no reply email. And then you'll have the attendee information that you can use to reach back out to them with their question.
Okay. Got it.
All right. Well, thank you very much for your time. We appreciate it. And again, if anything comes up, feel free to reach out. But if not, we will talk to you soon.
Okay, great. Thanks for your help.
Thank you. Bye-bye.